Bismillahirrahmanirrahim. Dear students, our today's lecture is about laboratory rearing of Trichogramma. The aim of this lesson is to learn the procedure for production of Trichogramma in the laboratory. The members of this genus are small and range in size from 0.2 to 1.5 mm. These parasitoids lay their eggs inside the eggs of other insects and are therefore used for insect pest control. Their main hosts are eggs of Lepidoptera, but they are also known to parasitize members of other insect orders like Hymenoptera, Neuroptera, and Coleoptera. Walking and short jumps are their mean of dispersal in field. Different Trichogramma species vary in their host specificity. There are more than 200 identified species of Trichogramma worldwide, and following are commonly reared species: Trichogramma chilonus, Trichogramma dendrolemi, and Trichogramma australiani, Trichogramma prettiosum. Trichogramma kills the insect pest at egg stage before they can cause any damage to crop. They live in a variety of habitats. They have been successfully released to manage the eggs of sugarcane borers, cutworms, cotton bollworms, and maize stem borers in different parts of the world. Use of Trichogramma in biological control programs. The major advantage of Trichogramma species as biocontrol agent of insect pests is their extremely small size, ease of rearing in the laboratory on artificial host eggs, and short life cycle. Due to their success in biological control program, 9 species of Trichogramma are produced commercially in insectaries around the world, with 60 countries leasing them. Trichogramma are used for control on numerous crops and plants. These include cotton, sugarcane, orchard, and forests. In Pakistan, they are being successfully used for the biological control of sugarcane pests. Briefly about their life cycle, the female Trichogramma lays eggs inside the host egg, which hatches inside, and after 3 to 4 days, the color of host egg changes to black, which indicates that the egg is parasitized, and after 4 to 5 days, adult emerges from the host egg. Now coming to the topic of producing Trichogramma in laboratory. For Trichogramma production in the laboratory, there are mainly 2 steps. Number 1 step is mass production of host eggs, and for host eggs, usually rice moth, Corsera cephalonica, or Cytotrega cerealela eggs are used. The number 2 step is the production of Trichogramma using laboratory produced host eggs. Now we will discuss the step number 1, which means host eggs. In this case, we are using rice moth, Corsera cephalonica eggs for production of host eggs. The grains of sorghum, millet, or maize can be used for rearing Corsera cephalonica in laboratory. The broken grains are heat sterilized to eliminate the residual population of stored product insects. Then 2 kg of broken grains are transferred to plastic cages along with 5 kg yeast to regulate larval growth. As you can see this in this plastic bottle, the grains are kept in this plastic bottle. Then Corsera cephalonica eggs are sprinkled on top of the grain, and you can see different life stages of the Corsera cephalonica are developing inside these grains. The Corsera eggs, after hatching and completing all larval stages, develop into adults, as you can see here in this picture. The adult Corsera cephalonica moths are then collected in the plastic boxes. The eggs are then collected from these boxes, as you can see here. The Coursera eggs are then pasted on the card with the help of dilute acacia gum. The eggs are sterilized by exposing the cards under 30-watt UV tube at a distance of 35 cm from the source for 10 minutes to kill their embryo. Following information should be provided on the card: name of the laboratory which is producing trichogramma, release instructions, date of preparation of cards, and expected date of parasite emergence. You can see the cards here are pasted, the eggs are pasted on these cards with the help of gum, and information about the laboratory and expected date of parasite emergence should be provided on these cards. These cards are then exposed to adult trichogramma for 24 hours at 28 to 25 degrees centigrade in transparent plastic containers. As you can see here, the trichogramma cards are placed inside these tubes, and then trichogramma female are released inside these tubes. The female trichogramma immediately start parasitizing the eggs of Coursera. Then step number 2, trichogramma production. Parasitized eggs start turning black on the 3rd day after parasitization. Life cycle of trichogramma is completed in 7 to 8 days. The eggs, when turned black, can be stored in the refrigerator or can be transported for field release. You can see here the eggs, black eggs, and the female trichogramma is emerging from the host egg. The emergence of adult generally takes place after 3 to 6 days after blackening of eggs. So this was all about trichogramma rearing in laboratory. If you have questions, you are more than welcome to ask questions, and thank you very much.